while people are still trickling in, let me start. And before I introduce today's speaker, let me just remind you of a couple of housekeeping items. We have the website of the DFHCC Connect Science Seminar that you see on the slide here, and that has all previous recorded lectures as well as a list of future lectures. So I urge you to check it out if you're interested in these seminars. I would also like to remind you that all questions should be posted using the Q&A tool and not the chat function. And we will try to get to as many of those at the end as possible, but we'll also pass on the ones we can't directly address to the speaker. Now, it's my great pleasure to introduce today's speaker for the virtual DFHCC Connect Science Seminar, James Altman, who joins us from UC Berkeley. James is an associate professor of molecular and cell biology at UC Berkeley, and he's also an investigator of the Chan Zuckerberg BioHub. And prior to joining the faculty at UC Berkeley in, I believe, 2013, he received a PhD in neuroscience at Emory and was then a postdoc with Ron Copito at Stanford, where I think he really developed his interest in the biology of lipid droplets and the homeostasis of lipid droplets. This continues to be the major focus of his work currently in his lab, and his lab has really provided seminal contributions to our understanding of the lipid droplet proteome, the biology of lipid droplets, and importantly, how these interact with other organelles and really serve as interorganelle communication pathways, if you want so, for major processes in all of our cells. This work provides important insights for a wide variety of therapeutic indications where these lipid droplets really go wrong. He has been awarded with a number of prizes for his groundbreaking work, including most recently the Günther Global Early Career Award and the Miller Institute Professorship Award. I don't want to take more time of his seminar, so please join me in welcoming James, and I'm really looking forward to his talk.
Okay, thank you for the very kind introduction and for the invitation. It's really a great pleasure to have this opportunity to share with you some of our research and ideas around the cell biology of lipid homeostasis, including some of the basic biology around lipid droplets and lipid metabolism, and how we've become interested in lipotoxic pathways and their relevance to cancer. So lipids in the cell can be stored in ER-derived neutral lipid storage organelles called lipid droplets. And for years, lipid droplets were actually largely ignored by cell biologists. The predominant thought was that they were simply inert globules of fat of little interest. We now recognize that lipid droplets are in fact highly dynamic organelles that are inherently connected with cellular energy and lipid metabolism. And so the research in my lab focuses on several aspects of lipid metabolism and lipid droplet biology. We're interested in how these organelles form and how their proteome is established and remodeled by the ubiquitin proteasome system. We're interested in how these organelles interact with other organelles and communicate lipids to these other organelles. And more broadly, we're interested in how the cell responds to lipid damage and how the cell prevents forms of lipid damage and lipotoxicity, such as the cell death process known as ferroptosis. So broadly speaking, my lab is interested in understanding two major questions, which we'll talk about today. How are lipids stored and used in cells? And how do cells respond to oxidative lipid damage? And can we begin to target this therapeutically for human benefit? So when we think about lipid droplets, we often think about adipose tissue and white adipocytes specifically. And indeed, this tissue and cell type are highly specialized for the storage of lipids and lipid droplets. You can see this in an artistic illustration here, as well as in a histological image from a mouse white adipocyte tissue. And you can see really large lipid droplets. These are up to 100 microns in diameter, nearly completely filling the cell, so enormous for a single organelle. But I want to point out that lipid droplets are highly conserved. You can see lipid droplets all the way from yeast through C. elegans, Drosophila, and into humans, and you can see lipid droplets in many tissues and cell types, such as muscle, hepatocytes in liver, and even astrocytes in the brain, as well as many other cell types. And so really the ability to form lipid droplets and the ability to store neutral lipids in lipid droplets is a conserved ubiquitous feature of nearly all cell types. Now, as you might expect, there are prevalent metabolic diseases associated with the aberrant accumulation of lipids and the deposition of those lipids into intracellular lipid droplets. And furthermore, there are mutations in genes encoding lipid droplet proteins and biogenesis factors that have been implicated in the etiology of metabolic diseases. These include diseases associated with too few lipid droplets, like lipodystrophy, cachexia, as well as diseases associated with too many lipid droplets, like fatty liver disease, atherosclerosis, obesity, and many others. Now, cancer cells are exposed to fluctuations in nutrient availability, and they exhibit remarkable metabolic plasticity, switching between alternative metabolic pathways to adapt and survive under different conditions. And lipid droplets function as cellular hubs of lipid metabolism, and they can really serve numerous functions that are relevant to cancer. Now, their formation can be induced in response to lipids from a wide variety of sources, such as through exogenous uptake of exogenous lipids, through de novo lipid synthesis, or the autophagic breakdown of membranes in the cell. And lipid droplets themselves can be broken down to provide building blocks that can influence energy metabolism, membrane homeostasis, and lipid signaling. And there are many ways that you can imagine lipid droplet biology could be exploited to promote cancer growth. And some of these functions that influence cancer progression include functioning as a nutrient storage depot for fatty acids, cholesterol, and vitamins, modulating membrane saturation and composition, and providing building blocks that can be used for membranes, energy, or the generation of lipid signaling molecules. And these are just a few examples of the connections between lipid droplets, cancer, and other diseases that underscore the importance of understanding the basic mechanisms that regulate lipid droplet biology. So the biology of lipid droplets and the formation of lipid droplets is an active area of investigation, and several steps have been identified in the formation of lipid droplets. This begins with the synthesis of neutral lipids like triacylglycerol, or TAG, which occurs through this cascade of acyltransferases, and the deposition of these neutral lipids between the leaflets of the ER. And at a high concentration, you have phase separation of the neutral lipid, forming this lipid droplet lens, which then emerges from the outer leaflet of the ER into the cytoplasm. And the lipid droplet can then continue to grow through local triacylglycerol synthesis, or in certain cell types through lipid droplet fusion. The cell can access stored lipids within the lipid droplet by the recruitment and activation of lipid droplet resident lipases, or through targeting of the lipid droplet through a selective autophagic pathway now known as lipophagy. Now, these organelles are unique. So in contrast to other organelles in the cell, they lack a phospholipid bilayer, and instead they consist of a single phospholipid monolayer that encircles a core of neutral lipids. This is mainly triacylglycerol and steryl esters, but they can also contain other hydrophobic molecules, such as fat-soluble vitamins and fat-soluble drugs, and they can influence the metabolism of these molecules as well. Now, the core of the lipid droplet is lipid-rich and does not contain any proteins, and instead the proteins are found decorating the phospholipid monolayer with the hydrophilic domains oriented towards the cytosol. Now, as I mentioned, these organelles function really as dynamic hubs of lipid metabolism, and so they play, as expected, broad roles in regulating energy, membrane biology, lipid signaling, and by sequestering lipids, they can also influence lipotoxicity in the cell. Now, all of these broad functions of lipid droplets are regulated by proteins that decorate the surface of the organelle. And these can be broadly classified into two types, depending on their trafficking pathway to the lipid droplet. These include class 1 lipid droplet proteins, which are insert first into the ER, laterally diffuse in the outer leaflet of the ER into the forming lipid droplet, and class 2 lipid droplet proteins, which are thought to insert directly into the lipid droplet. Now, given the importance of the proteins for regulating lipid droplet biogenesis, lipid droplet functions, and cellular lipid metabolism, we and others have been very interested in the composition of the droplet proteome. So what is the composition of the droplet proteome? And perhaps more challengingly, how is this composition established and how is it regulated and remodeled in different cell types and under different metabolic conditions? And of course, we're interested in defining the functions of the various lipid droplet proteins and their importance in cellular lipid and energy metabolism. So I'll touch on some of our recent findings that address each of these questions. So the idea of defining a lipid droplet proteome composition is not a new question, and this has been addressed before using biochemical fractionation approaches in which your favorite cell type is down homogenized, broken, layered under a sucrose gradient, and after a short spin, the lipid droplets will float into this buoyant fraction, and this can be isolated for mass spec and proteomic analysis. And this is just shown here that lipid droplets are buoyant due to their fat composition and can be floated to the top of a gradient. However, while this has identified bona fide lipid droplet proteins, this method has been challenging to interpret due to high numbers of false positives. And this is because the lipid droplets in the cell don't exist in a vacuum. They have multiple organelle-organelle interactions, and some of those organelles and membrane fragments come along for the ride during this fractionation step. So for example, ER proteins are abundant contaminants in this fraction. And so given this obstacle, two members of the lab, a postdoctoral fellow, Cheryl Brazuker, and a graduate student, Clark Peterson, sought to define a more high confidence method to understand the droplet proteome composition. And in this approach, they utilized a proximity labeling method first developed by Alice Ting's lab that probably many of you are familiar with. Very briefly, we target an engineered ascorbate peroxidase, APEX2, to the surface of lipid droplets. And then this enzyme can tag neighboring proteins with biotin when incubated with biotin phenol and a short pulse of hydrogen peroxide. Essentially, what this allows us to do is to selectively tag lipid droplet proteins with a chemical moiety that we can then exploit for the isolation of these proteins for proteomic analysis. So using this method, we defined very specific high confidence lipid droplet proteomes in multiple human cancer cell lines, UTOS, an osteosarcoma cell line, and HA7, a hepatocellular carcinoma cell line. And we're just providing a snapshot here that's organized into functional modules. And I'll just point out a few salient features. First, we identified, as anticipated, many proteins involved in triacylglycerol and lipid metabolism. And these included the perilipin family members, PLIN2 and PLIN3, which are canonical lipid droplet localized proteins, acyl transferases like GPAT3 and GPAT4, acyl-CoA synthetases, lipases, and their regulators. We also identified proteins belonging to functional classes that are not directly related to lipid biology, such as ubiquitination. And several of these proteins are known to be involved in targeting proteins from the early secretory pathway to the proteasome through a process known as ER-associated degradation, or ERAD. And the presence of these proteins on the droplet led us to hypothesize a functional connection between ERAD and lipid droplet protein degradation in proteome composition. And we can imagine a few ways that this could happen. We know that many lipid droplet proteins insert into the ER and then laterally diffuse in the outer leaflet into the lipid droplet. And so we could imagine that these class 1 lipid droplet proteins might be subject to regulation through canonical ERAD complexes, where they're first ubiquitinated, extracted by the AAA ATPase, VCP, to be targeted to the proteasome. We can also imagine a parallel pathway utilizing lipid droplet localized ubiquitination machinery, in which these substrates are similarly ubiquitinated and extracted for proteasomal clearance. And these two pathways, of course, aren't mutually exclusive to one another. So to understand the mechanisms that degrade lipid droplet proteins, we really needed substrates that we could use as tools to begin to dissect these pathways. And we reasoned that the AAA ATPase VCP is a convergent factor in both of these pathways. And then if we inhibit VCP, we could accumulate substrates that are being degraded by either of these pathways and track these substrates in a manner that we could then identify using our proteomic approaches. And so we devised a quantitative proteomics approach in which we used SILAC labeling. So we heavily labeled one cell population and treated this population with CB5083, a specific inhibitor of the AAA ATPase VCP. And then we subjected these cells to our proximity labeling proteomics approach. Under these conditions, these were oleate-treated UHOS cells. Most lipid droplet proteins were unchanged, the levels, so a 1-to-1 distribution. And we saw that one protein accumulated in response to VCP inhibition, and this was a protein called C18orf32. We have data indicating that C18orf32 likely is regulating the lipid composition of lipid droplets, but I'm going to focus on C18orf32 as a tool to understand how lipid droplet proteins are degraded. Now, briefly, C18orf32 seems to exhibit properties of a class 1 lipid droplet protein. So it localizes to the endoplasmic reticulum under basal conditions. You can see that here in these time-lapse movies. It shows a very reticular distribution. We could see that small puncta that seem to be zipping along this reticular network, which we reasoned were likely to be nascent lipid droplets still associated with the ER. And this seems to be the case. So we see even at early time points, these puncta label with fluorescently labeled fatty acids. And these puncta also label with early markers of lipid droplet biogenesis, including these genetic markers, the HPOS and live drop, which label these early still ER-associated lipid droplets. In addition, if we treat these cells with the fatty acid oleate to induce lipid droplet biogenesis, we see a dramatic change in C18orf32 distribution, moving from the endoplasmic reticulum out onto these lipid droplets. And indeed, we could mark these lipid droplets with a stain. These are, in fact, as we anticipated, are indeed lipid droplets. So what I told you is interesting about this protein is that it's potentially being degraded by the proteasome, and we could use this to understand how lipid droplet proteins are cleared and regulated. And indeed, this is the case. That has a half-life of about 4 to 5 hours, and this degradation is completely blocked by inhibitors of the E1 ubiquitin activating enzyme, inhibitors of the proteasome, and inhibitors of the AAA ATPase VCP. Now, we wanted to understand the mechanism that targets this protein to the proteasome, and specifically, we really wanted to understand the E3 ligase that mediates the ubiquination of this protein. And so we used another proteomic approach in which we affinity purified C18orf32 and looked at the interacting proteins, and very nicely we saw that it associated with VCP and ubiquitin under these conditions. And we also saw proteins that have been implicated in canonical ERAD pathways. Most notably is this protein DRLIN1, which is one of the best characterized proteins involved in ERAD. This is a polytopic ER-resident membrane protein. It's a rhomboid pseudoprotease that's involved in the dislocation or retrotranslocation of membrane proteins from the ER into the cytosol. And indeed, when we knock out DRLIN1, we see this marked stabilization in this protein. So it's reducing the degradation of this protein. And while we didn't identify the E3 ligase in this proteomics experiment, we then performed a candidate-based screen for ERAD E3 ligases. And we found that knockout of GP78, a well-known E3 ligase, also similarly stabilizes C18orf32. Knockout of other E3 ligases like RNF5 or HERG1 had no effect. And importantly, when we knock out GP78, we see a small increase in C18orf32 in the ER, but we see a much larger increase of C18orf32 in the lipid droplet enriched buoyant fraction. What these data tell us is that C18orf32 is a class 1 lipid droplet protein migrating between these two organelles, and that its levels are being regulated by this canonical ERAD pathway mediated by GP78, DRLIN1, and VCP. And when we knock out this pathway, we accumulate C18orf32, allowing more protein to traffic and flux into the lipid droplet. So this really highlights the high degree of crosstalk occurring between these two organelles. Now, this made sense for a class 1 lipid droplet protein, which localizes first to the ER. But a graduate student in the lab, Melissa Roberts, was really interested in understanding how a class 2 lipid droplet protein would be regulated. Something like perilipin-2, which is thought to insert directly into the lipid droplet independent of an ER intermediate. So how are these proteins degraded? Are they degraded through a unique proteasomal pathway, maybe directly off the lipid droplet, or do they somehow access this ERAD-based mechanism? So in order to understand how class 2 lipid droplet proteins are degraded, Melissa decided to study PLIN2. And this is a lipid droplet scaffold protein. Its expression is known to stabilize lipid droplets. And in fact, its levels are known to be regulated in part by degradation by the proteasome, though the mechanism remains mostly unclear. This is also a functionally important protein, so knockout of PLIN2 in mice protects against diet-induced fatty liver, and their mutations or polymorphisms associated with altered triacylglycerol metabolism in humans. So to understand how PLIN2 is being regulated, Melissa decided to take a genetic approach, and she first developed a fluorescence-based reporter cell line in which she inserted GFP using CRISPR into the endogenous locus. This is endogenously expressed PLIN2 tagged with GFP. This is actually a heterozygous line, so we see that PLIN2 has been modified with GFP, but we also see an untagged endogenous form of PLIN2, which actually serves as a very nice internal control for our experiments. And we see very nice localization of PLIN2 GFP to the surface of lipid droplets by fluorescence microscopy. Now, nicely, this fluorescence-based reporter cell line allows us to study PLIN2 both by fluorescence microscopy and also by fluorescence, by flow cytometry and fluorescence activated cell sorting. And so we see here that has a nice peak of GFP fluorescence. If we induce the lipolytic consumption of the lipid droplet, we see a decrease in the GFP fluorescence as PLIN2 is being targeted to the proteasome for clearance. And indeed, if we add MG132, an inhibitor of the proteasome, we maintain the levels of GFP, indicating we blocked the proteasomal clearance of PLIN2 under these conditions. So this reporter cell line in hand, Melissa decided to take a CRISPR genetic approach to understand how it's being degraded and to tease out the proteins involved in this degradation. So she took her reporter cell lines, and these are HA7 hepatoma cells, and she infected them with a ubiquitination-enriched sgRNA library. She could then, by fluorescence activated cell sorting, simply sort for low and high GFP fluorescent cells. So the cells that exhibit a decrease in PLIN2 GFP degradation would be predicted to be present in this high fluorescent gate. And then she could deconvolve these data by deep sequencing. So here I'm showing you the data from this genetic screen as a volcano plot. And Melissa identified several interesting ubiquitination components in this screen, including the E2 ubiquitin conjugating enzyme UBE2J2, the E3 enzyme MARCH6, and the E3 or E4 UBR4. And she could validate these data by generating multiple knockouts using independent guides against each of these genes. So knockout of MARCH6, UB2J2 or UBR4 results in a partial stabilization of PLIN2. So you can see this rightward shift in the histogram of PLIN2 GFP. And this was really interesting because we know that all of these components cooperate together in a known pathway. So MARCH6 and UB2J2 are integral ER resident membrane proteins that are known to functionally and physically associate. And UBR4, while it's more broadly involved in the degradation of cytosolic proteins, it's been recently shown to act downstream of MARCH6 and UBE2J2 in ERAD pathways, where it mediates the conjugation of branched ubiquitin chains to these substrates, presumably thereby increasing the efficiency of proteasomal targeting for these substrates. So it's really interesting that all three of these components came out in this genetic screen. We've gone on to do additional validation of MARCH6. So in these MARCH6 knockout cells, you can see a marked stabilization of PLIN2, GFP, and the endogenous protein when we induce lipolytic degradation of the droplet. We actually see increase in steady state levels of PLIN2. So this is PLIN2 GFP, as well as the untagged PLIN2 that's present in these cells. And we see a partial stabilization in the turnover during lipolytic degradation of the droplet. And this is measured by flow cytometry. So there's likely additional pathways that are compensating for loss of MARCH6, given that this is a partial stabilization. Now, MARCH6 has a well-characterized substrate that's known to be involved in lipid metabolism. So it degrades squalene monooxygenase, SQLE, which is plays an important role in cholesterol synthesis. And so we wanted to determine whether or not this was relevant to the stabilization of PLIN2. Perhaps it's indirectly affecting PLIN2 levels by regulating cholesterol metabolism. So we also looked at squalene monooxygenase degradation and depleted SQLE by siRNAs. So as expected in our MARCH6 knockouts, they have a marked stabilization in SQLE. But when we deplete SQLE and return its levels to basal levels, this had no effect on the accumulation of PLIN2GFP or the untagged PLIN2 present in these cells, indicating this independent of this SQLE stabilization. And we also looked to see if this impacted neutral lipid storage, and indeed, these MARCH6 knockout cells had an increase in lipid droplets under all conditions, so under vehicle-treated conditions, under Trixin-treated conditions inducing lipolysis, and under oleate-stimulated conditions. This was quite dramatic, the increase in lipid droplet by amount in these cells. We could also measure this by flow cytometry using a neutral lipid stain. And so you see this rightward shift in the histogram. And again, the knockdown of SQLE had no effect on this accumulation in these MARCH6 knockouts of lipid droplets and neutral lipids. Again, pointing to this effect being independent of its substrate SQLE. So there's a lot of questions that remain, but both our proteomic approaches and our functional genomic approaches really point to an intimate relationship between lipid droplet proteome composition regulation and these ERAD pathways. I think it's too early to say this as a rule for lipid droplet proteins. I think there are likely to be cytosolic degradation pathways that contribute, as well as potentially lipid droplet associated degradation pathways. So we identified ubiquitination components on the lipid droplet, but we still don't understand the function of these components, whether they're playing a role in a proteasomal pathway or a non-proteasome ubiquitin regulatory pathway. And of course, we're very interested in understanding how these complex pathways regulate lipid droplet proteomes under different metabolic conditions and broadly contribute to lipid metabolism regulation. So in addition to these ubiquitin-related proteins found on lipid droplets, we're also interested in other functional modules that we had identified on lipid droplets. And one functional module that really caught our attention contained proteins with annotated roles in redox activities. And we become very interested in a protein that's now known as FSP1. Its gene name and former name is AIFM2. And in this paper we had published a few years ago, we had indeed found that FSP1 GFP localizes very nicely, at least in part, to lipid droplets as well as to some other membranes. But the function of this protein was a mystery for us at this time. So we had knocked out FSP1, and we didn't see large effects on lipid droplet amount or lipid droplet biogenesis or turnover kinetics. But recently, a postdoc in the lab made a breakthrough by connecting FSP1 to oxidative lipid damage and a cell death process known as ferroptosis. So I'm going to briefly introduce ferroptosis, and I'll tell you about the functions we've discovered for FSP1 in this process. So ferroptosis is a form of regulated cell death. It's distinct from other forms of cell death like apoptosis, and it involves the iron-dependent accumulation of oxidatively damaged lipids. So polyunsaturated fatty acids are incorporated into phospholipids, and these are particularly sensitive to this iron-dependent oxidative damage in the accumulation of these lipid peroxides. Now, if these accumulate to high levels, the membrane eventually ruptures and the cells die by ferroptosis. Now, there's a well-known pathway that mediates and protects cells against ferroptosis, and this is mediated by a glutathione-dependent peroxidase, GPX4, which corrects these lipid peroxides into non-toxic lipid alcohols. And this process requires glutathione, so it's sensitive to inhibitors of the system Xe- channel, Iraxin. And this channel exchanges glutamate for cysteine, which is required for the production of glutathione. And there are also direct inhibitors of GPX4 like RSL3, which we use widely in our studies. And this pathway has gained a lot of attention because oxidatively damaged lipids are known to accumulate in many pathological degenerative conditions. And there's been also a lot of excitement about whether this pathway could be targeted as a way to treat cancer. And indeed, there have been many studies targeting GPX4 in these pathways that have shown promise in preclinical models. But it's also been noted that some cancer cells are resistant to GPX4 inhibitors. And this was something that really caught our attention suggested that there were potentially additional protective pathways. And we were very interested in this question, both from the basic science perspective that we might be able to gain some additional understanding to how oxidative lipid damage is regulated, as well as the therapeutic perspective that maybe these additional pathways are mediating cancer resistance against ferroptosis in these drugs and could be important targets to consider for additional cancer therapeutic approaches. And so this was a project that was tackled by a team in the lab. Again, this terrific postdoc, Cheryl Berzuker, graduate students Joe Hendricks and Melissa Roberts, and another postdoc, Zhipeng Li. And so they sought to understand the mechanisms that mediate cancer resistance to these GPX4 inhibitors in ferroptosis. And again, they turned to genetics and they performed synthetic lethal CRISPR screens in collaboration with Mike Bassick's lab down at Stanford. And in this experiment, the osteosarcoma cell line UTOS, which are resistant to RSL3 and other GPX4 inhibitors, were infected with a genome-wide sgRNA library, so 10 guides. And then these were simply treated either with a control treatment, a vehicle treatment, or a sublethal dose of RSL3, our GPX4 inhibitor. So under these conditions, our normal control UTOS grow normally, but we'd anticipate if they're lacking a key factor that mediates resistance against ferroptosis, that these would die, be disenriched in the final population, which we could then identify by deep sequencing. And so here I'm showing you the data as a volcano plot. And you can see that one gene really stood out as having a high gene effect and a high confidence score. And lo and behold, this was FSP1, formerly known as AFM2, but we renamed this protein ferroptosis suppressor protein 1 to reflect its function in cells. And you can really appreciate the power of these genetic screens here in this cloud plot. Shown in gray are the thousands of controls sgRNAs present in this library. And then you can see that 9 of the 10 sgRNAs targeting FSP1 were disenriched in that final population, which is why we had such a high confidence score in this gene. Now, I also want to point out that this was co-discovered at the same time by Marcus Conrad and Conrad's group and Pedro Angeli, and these papers were simultaneously published. And very nicely, they used a very complementary systems level approach, doing an overexpression suppression screen. So they took a lentiviral cDNA library, infected a sensitive line, MCF7 cDNAs into a PFA1 cell. So these are a sensitive line to ferroptosis. And then they looked for genes that, when overexpressed, suppress this ferroptosis. And very nicely, they identified 2 genes, GPX4 and FSP1. So our findings are very consistent. And so we've gone on to validate these findings, of course. We generated FSP1 knockout cells in the UTOS. And you can see here that the control cells grow fine when incubated with RSL3, but these FSP1 knockout cells exhibit a classic characteristic ferroptotic morphology and are marked here by a cell death marker. We can perform dose response analyses of RSL3 induced death. So our control cells are highly resistant even to high doses of RSL3, where you can see our 2 knockout cell lines generated with independent guides here are highly sensitized with an EC50 around 50 to 100 nanomolar. So this is indeed ferroptosis. So we can block the death using known inhibitors of ferroptosis like DFO, a chelator of iron, as well as FER1, ferrostatin, and adiponone, which are lipophilic antioxidants. So here again is our knockout, and these are blocked. Their death and resistance is gained again by adding these inhibitors of ferroptosis, but not inhibitors of other cell death pathways. Similarly, we know that ferroptosis is exquisitely sensitive to the presence of polyunsaturated fatty acids in the membrane, and these require the acyl-CoA synthetase ACSL4 for activation and subsequent incorporation into phospholipids. And if you knock out ACSL4, these cells become resistant to ferroptosis. And indeed, if we generate a double knockout of FSP1 and ACSL4, you see we gain this really remarkable resistance to RSL3, contrasting our FSP1 knockout. And so we wanted to understand more about the cell biology of FSP1 and the mechanism by which it's contributing to this resistance to ferroptosis. So we first looked again at the localization of FSP1. And in these experiments, we knocked in a halo tag into the genomic locus of FSP1. This is endogenously expressed FSP1 halo. And then very nicely, again, we see very nice lipid droplet localization of FSP1. So these nice circular structures marked with BODIPI. And we also see localization to some other membranes, particularly the plasma membrane, which is shown here in these ruffles, marked by cell mask. And we could confirm this plasma membrane localization both by turf microscopy as well as by separation of these fractions across iodoxanol gradients. And so to understand whether this membrane recruitment was important, we really needed to understand more about how FSP1 is recruited and targeted to these different membranes. And as I mentioned in the first half of my talk, there are different classes of lipid droplet proteins. Some insert into the ER and localize to droplets, and some are recruited as class 2 lipid droplet proteins. When we looked at FSP1, it consists mainly of an oxidoreductase domain, which I'll come back to, but also had at the N-terminus a myristoylation consensus sequence. And this suggested that perhaps FSP1 is acting like one of these class 2 lipid droplet proteins, being myristoylated and then recruited to lipid droplets and anchored there by this myristate. And so we first sought to determine whether or not indeed FSP1 is myristoylated. We used a click chemistry approach to do this, in which we metabolically label our cells with an alkyne modified myristate. And then after lysis, we can use click chemistry to add molecules with useful properties, such as the fluorescence camera for in-gel fluorescence or a biotin that we can use to affinity purify these myristoylated proteins. So here we've performed this approach and then affinity purified myristoylated proteins from lipid droplets. And nicely, you can see that the endogenous FSP1 readily co-precipitates, indicating that it is indeed myristoylated. We'd anticipate that this would be myristoylated at the glycine at position 2. So canonically, proteins are co-translationally myristoylated after removal of this initiator methionine. So we generated a GFP tagged G2A mutant, which we mutated that glycine 2 to an alanine. And very nicely, you can see that the wild type protein is still myristoylated, but this G2A mutant is no longer myristoylated. And we can lose the myristylation of both GFP and endogenous FSP1 simply by incubating with an N-myristyltransferase inhibitor. And indeed, this is co-translationally myristoylated because we can block this with an inhibitor of translation called emetine. This seems to be myristoylated through the canonical pathway. Now we have some tools to begin to study its trafficking. So we again looked at its trafficking using fluorescence microscopy. We see nice lipid droplet localization. But now if we add this myristylation N-myristyltransferase inhibitor or the G2A mutant, we largely see a diffuse cytosolic distribution and very little lipid droplet localization. We also lose the plasma membrane localization of the G2A mutant, which we quantified by biochemical fractionation experiments. Importantly, now we can ask if this membrane recruitment is important for its role in ferroptosis, and indeed it is. So here you can see our sensitive knockout cells. If we introduce FSP1 wild type, this rescues ferroptosis resistance. But if we introduce our G2A mutant, which is actually expressed at higher levels than wild type, has no effect on ferroptosis. So clearly FSP1 is localizing to multiple membranes, to the plasma membrane, into the lipid droplet, and somehow it's inhibiting ferroptosis. Now what this experiment didn't tell us was whether or not FSP1 recruitment to one or both of these membrane compartments was important for ferroptosis resistance. And to answer this, we generated a series of FSP1 fusion proteins that are targeted to different compartments in the cell. So we fused FSP1, for example, to signal sequences that targeted to the outer membrane of the mitochondria, to the lipid droplet, to the plasma membrane, or to the outer leaflet of the ER. And then we could ask whether or not when FSP1 is targeted to these discrete subcellular compartments, is this sufficient to rescue ferroptosis resistance? So again, perform these rescue experiments. And what was interesting was we only saw rescue with wild type FSP1 or the plasma membrane targeted FSP1. Our lipid droplet targeted FSP1 or FSP1 targeted to these other membranes had no effect on ferroptosis resistance. So it didn't appear that lipid droplets, at least in this scenario, I think this is probably context specific, were important for ferroptosis resistance. And we did a few other experiments to convince ourselves of this fact. For example, we depleted lipid droplets using inhibitors of diacylglycerol acyltransferases 1 or 2, which are required for triacylglycerol synthesis. And this largely depletes lipid droplets in these cells. And we could see that under these conditions, when we deplete lipid droplets, this really had no effect on ferroptosis resistance, really suggesting that, at least in this context, lipid droplets weren't important for ferroptosis resistance or for FSP1 function. And then FSP1 is acting at the plasma membrane to inhibit ferroptosis. Now, we'd anticipate, if this was the case, that FSP1 would be impacting the accumulation of these lipid peroxides, which is a very downstream step in ferroptosis. And this experiment to test this was done in collaboration with our collaborator, Scott Dixon, down at Stanford. And here we're using a fluorescence reporter of lipid peroxidation. This is Bodipi C11, which fluoresces green in response to conditions that oxidize PUFAs in the accumulation of lipid peroxides as an indirect measure. And what you can see is the FSP1 knockout cell lines are highly sensitive to the accumulation of lipid peroxides under this RSL3 treatment. You can see a quantification here that they really accumulate high levels of lipid peroxides in response to RSL3. So we were then interested in the mechanism. How is FSP1 actually blocking the accumulation of these lipid peroxides? We first measured glutathione just to be sure there was no change in glutathione uptake in this pathway. Indeed, there didn't seem to be any effect. In fact, there may have been a slight trend towards increased glutathione under FSP1 knockout, so this wasn't significant. And you can see here the control with Avastin, which blocks the system Fc- channel, completely depletes glutathione under these conditions. We also looked at the incorporation of PUFAs into membranes. So we considered the possibility that perhaps there's increased PUFAs present in the membranes that could sensitize these cells to lipid peroxidation and ferroptosis. So these lipidomics experiments were in collaboration with Dan Nomura here at UC Berkeley. And what we actually saw was there's a decrease in PUFA-containing lipids and an increase in lysophospholipid. And some of the quantification is shown here, where you accumulate lysophospholipid in multiple FSP1 knockout cell lines. So we certainly weren't accumulating PUFAs in the membranes. And this is actually, this accumulation of lysophospholipids in the membranes is a lipidomic signature of ferroptosis that other researchers have seen, likely due to phospholipases that are removing oxidized phospholipids from the SN2 position. These data and other data led us to consider the possibility that FSP1 was acting through a non-canonical mechanism to suppress ferroptosis. So as I mentioned, FSP1 has this oxidoreductase domain, and it had been previously shown, at least in vitro, that FSP1 could reduce this lipid molecule coenzyme Q10 or CoQ, also known as ubiquinone or ubiquinol. And this is a really interesting lipid. It has this very oily hydrophobic polyisoprenoid tail, and it has this redox active head group, which allows it to move electrons and to act as a lipophilic antioxidant, which was interesting to our group. But when we thought about coenzyme Q10 and CoQ, we thought about the mitochondria, because this is where it plays its most famous role within the electron transport chain. And we never seen FSP1 in the mitochondria. We'd only seen it at the plasma membrane and lipid droplets. So we began to wonder if CoQ was relevant or whether this might be just an in vitro artifact. But when we began to dig into the literature, we began to appreciate that CoQ has actually been known to be present in non-mitochondrial membranes. And this has actually been known for a long time. So CoQ and its role in the mitochondria was discovered in really beautiful biochemistry work back in the 1950s. And as early as 1961, it was shown that CoQ in fractions is present in non-mitochondrial membranes. But its real function in these non-mitochondrial membranes has remained a mystery for the last 60 years. So our data led us to a hypothesis that perhaps FSP1 is acting as to reduce non-mitochondrial CoQ, and that way generating a lipophilic antioxidant locally that can suppress the propagation of lipid peroxides and ferroptosis. So we began to test this model. We generated an oxidoreductase dead mutant of FSP1 mutating E156 to an alanine. We first tested the activity of this protein in vitro using purified protein from bacteria, and you can see that we had really nice activity through a model substrate, Rosazurin, as well as to CoQ in vitro, and this was abolished by this E156A mutation. Importantly, when we introduce wild-type FSP1 in FSP1 knockout cells, we increase the amount of reduced CoQ in these cells. When we introduce this E156A mutant, this has no effect relative to our knockouts. And this was measured by mass spectrometry in collaboration with Peter Tang's group. And this is probably an underestimation of the effects FSP1 is having on non-mitochondrial CoQ, because these are done from whole cell lysates. So looking at mitochondrial and non-mitochondrial pools of CoQ. But this was an important demonstration that FSP1 is indeed functioning as a CoQ oxidative reductase in cells. We could then take this mutant and perform rescue experiments to examine the cancer resistance to ferroptosis inducers. And indeed, again, we see this rescue by our wild type FSP1, but this E156A mutant, which localizes normally in cells, has no effect on ferroptosis resistance. We could also see this when we looked at lipid peroxidation. So these FSP1 knockout cells, again, very sensitive to the accumulation of these lipid peroxides, reported here in this green color with GODIP-C11. And we can completely suppress this by adding back adibinone, which is a soluble mimic of CoQ. And this was also sufficient to suppress ferroptosis in these cell lines. Now, if this is indeed the case, if FSP1 is acting through non-mitochondrial CoQ to affect ferroptosis, we'd anticipate that simply depleting CoQ would have similar effects on ferroptosis. And so we generated CoQ2 knockouts. So this blocks the endogenous de novo synthesis pathway of CoQ. These cells still have about 50% CoQ levels because they gain CoQ from the serum, the FBS that these cells are grown in. But even this depletion of CoQ was sufficient to phenocopy our FSP1 knockouts and sensitize these cells to RSL3. And again, this type of cell death was blocked by FAIR1, a ferroptosis inhibitor. We also saw the same effect with lipid peroxidation. These CoQ2 knockout cells are sensitive to the accumulation of lipid peroxides, and this could be blocked by adding adibinone, the soluble mimic of CoQ. And so this is really interesting. It suggests this model in which FSP1 is acting through non-mitochondrial CoQ to suppress ferroptosis. Now, we're really interested in the biology of FSP1 and its relationship to cancer. I'm sure many of you are familiar with the Cancer Therapeutics Response Portal, which is a really beautiful database from Stuart Schreiber's group in the Broad that looked at the correlation of gene expression with many different small molecules and their ability to induce cell death. And we're very happy to see that FSP1 came up very strongly in positively correlating across nearly 1,000 cancer cell lines with resistance against 3 different GPX4 inhibitors, RSL3, ML162, and ML210. And this is the most highly correlated gene with resistance against GPX4 inhibitors in the genome. And we can validate this. We took low expressing lung cancer cell lines and high expressing lung cancer cell lines. We can see that these low expressors are highly sensitive to RSL3. These high expressing FSP1 cancer cell lines are very resistant to RSL3 induced death. And we can also make the expected manipulations. We can overexpress FSP1 in these low expressing cell lines, and we gain a large amount of resistance against RSL3, indicating that FSP1 is sufficient to induce this resistance. And we can also make knockouts in these very high expressing and very resistant cell lines, and we gain a remarkable nearly 3 orders of magnitude sensitization to GPX4 inhibitors in these cell lines. So I've shown you data using small molecules. We can also show this relationship using genetics. So we generated GPX4 knockouts and GPX4 double knockouts with FSP1 in these H460 cell lines. The GPX4, these cell lines had to be maintained in ferrostatin, the inhibitor of FAIR1. And then we did washout experiments where we removed FAIR1. And what you see here is that the GPX4 knockouts continue to grow normally, so no cell death, but our double knockouts where we've removed FSP1 rapidly die under these conditions. and through a ferroptosis mechanism that can be blocked by FAIR1. We can also show this relationship in tumor xenograft experiments done in collaboration with Dan Nomura here at Berkeley. Our GPX4 knockouts grow normally, even after induction of ferroptosis. This was done by removal of FAIR1 from these animals. But we see a marked decrease in the rate of tumor growth when we make a double delete of GPX4 and FSP1 together. Similar to this GPX4 knockout, when we just delete FSP1 alone, there's no effect on tumor growth. And so this indicates, again, this compensatory relationship between GPX4 and FSP1, both in our in vitro experiments and in this tumor xenograft experiment. So to conclude, what we've discovered here is, in addition to this glutathione-dependent pathway, there's an additional glutathione-independent pathway that centers around FSP1. And so using our genetic approaches, we have identified FSP1 as this independent pathway. And the mechanism of FSP1 is through the reduction of CoQ, which acts to suppress lipid peroxide propagation and subsequently ferroptosis. And so specifically acting at the plasma membrane. So I think this is interesting from a ferroptosis perspective, as this is an independent protective pathway. It's also interesting from a very basic perspective, because it suggests that the function of non-mitochondrial CoQ, which has been a mystery for decades, really is to maintain the quality of membranes and to prevent lipid oxidation to these membranes. And this also identifies FSP1 as a high priority target for the development of novel chemotherapeutics, which we could imagine would be used combinatorially with therapeutics targeting the glutathione-dependent ferroptosis pathway. So in conclusion, I've told you two stories here, one around how lipid droplet proteomes are remodeled under different conditions, both using a proteomic chemical biology proximity labeling approach, as well as using a new unpublished study in which we used a fluorescence-based reporter approach. And in the second portion, I told you about some of our studies using genetic platforms to understand aspects of cancer drug resistance, and how we use this to understand ferroptosis and reveal a new facet of ferroptosis and lipid quality control. So I'll just end here by thanking the members of my lab, particularly Kirill Berzuker, who really spearheaded both of these studies, and Melissa Roberts, who developed the fluorescence-based reporter approach to understand PLIN2 regulation. We've had a real terrific group of collaborators on these studies that I've tried to point out along the way, and also thank our funding sources for all these studies. And thank you, and I'm happy to take any questions at this time.
I wasn't able to unmute myself. Thank you, James, for this great talk. I have a question that's very general to start, and then I go to the Q&A. Is the size of these lipid droplets in some form regulated? Are there mechanisms at play that limit size? Do we know anything about that?
Yeah, that's a really great question. And you'll see that in some of the cell types, the size will differ dramatically. For example, in HeLa cells, they tend to have lots of small lipid droplets. In our Hus7 cells, they have very diverse in size, ranging from very small, 100, 200 nanometers to 3, 4 microns in diameter. And that's a good question where it's incompletely understood, but what I can say is that in certain cell types, they express proteins that mediate fusion. And so this is one way that lipid droplets can grow. So there are these side proteins which sit at lipid droplet contacts, which enable lipid droplets to fuse. Under other conditions, there seem to be subpopulations that grow and expand. So maybe through local triacylglycerol synthesis or through lipid synthesis in the ER that's delivered to these lipid droplets and allow them to grow. In addition, the actual phospholipids are important on the droplet. So if you don't have enough phospholipids to cover the droplet, as a surfactant, lipid droplets will start to spontaneously fuse, and you'll get very large lipid droplets as well.
Okay. We have a question here from Joyce Kanevsky. Is lipid droplet excess toxic to cells?
Yeah, I would say this is probably context specific. So under normal conditions, I think that lipid droplets are protective, and that and there's lots of evidence from our lab and others that if you block lipid droplet biogenesis under many conditions, this results in lipotoxic damage. For example, we've shown under a certain condition, if you block lipid droplets, you get the accumulation of acylcarnitines and you get mitochondrial dysfunction. And other groups have shown if you block lipid droplets under certain conditions, you get ER stress. So they really function as like a lipid buffer, sequestering these lipids and preventing some of the toxicity. Now, whether they could be toxic under certain conditions, that's a great question. You know, I think that under pathological conditions like fatty liver, you get these accumulations of really large lipid droplets. And so whether some of the toxicity there is due to these large lipid droplets, which might start associating with other organelles or non-specifically recruiting certain proteins and causing dysfunction, that's possible. It's also possible that these large lipid droplets are no longer sufficiently sequestering lipids, and so they're allowing lipotoxicity to occur. So I think there are multiple ways in which that could be the case under pathological conditions.
We have another question from Venkateshwar Krishamuni. Fantastic talk. Do you know what regulates FSP1 expression in cancer cells?
Yeah, that's a really good question. We're very interested in answering this question, and particularly because we see this really dramatic correlation in FSP1 levels with cancer resistance to ferroptosis. There had been some data suggesting that maybe P53 was regulating FSP1 expression, but we haven't seen this, at least in our studies. When we look, and maybe this is dependent on the cancer cell line. In the cell lines we looked at, we didn't see a role for P53 in regulating its expression, but perhaps this is in other cancer cell lines, P53 could be a regulator. But I think that this question is really an unanswered question that still needs to be addressed.
Maybe direct follow-up from me. Is it a stress responsive gene generally? Does it go up in response to certain stresses?
Yeah, at least not that we've seen. We've mostly looked at oxidative stressors and ferroptosis inducers, and we haven't seen induction in FSP1, at least in response to those stresses. And we also haven't seen dramatic changes in localization. So that was something we also looked at in some detail, whether or not, maybe when we're inducing ferroptosis, there's a relocalization of FSP1 to the plasma membrane, or recruitment or dynamic exchanges between the compartments. And we didn't see that, at least under the conditions that we looked at, though I think that remains a possibility.
Yeah. And we have another question from Kenichi Himada. Relevant to an earlier question, but did you see any correlation of the lipid droplet abundance and ferroptosis sensitivity across multiple cell lines?
Yeah, I don't think we've looked at that question in a detailed enough way to make a conclusion. We looked at it very carefully for FSP1 in the cell lines we were working on, and if we deplete droplets in the cell lines we were working, we didn't see an effect on ferroptosis. But I think it's very likely that lipid droplets in other contexts do impact ferroptosis. And there's some nice data from Stuart Schreiber's group in Yilong that shows at least in some contexts, like clear cell renal cell carcinoma, there's a relationship there between lipid droplets and ferroptosis sensitivity. And so they were showing HIF2 alpha induction of HILPTA, or HIG2, which is a lipid droplet localized protein that's known to inhibit lipolysis. And so you can imagine a lot of ways in which lipid droplets could influence, for example, PUFA sequestration or PUFA flux into phospholipids. And this could have a big effect on ferroptosis sensitivity. So I think that still remains an open question of how big a contribution that is. But I think it's likely to be the case that lipid droplets will end up impacting ferroptosis in different ways.
And one final question, and then we're at the hour. Do you know what dictates lipid accumulation, droplet formation versus fatty acids being shuttled to other organelles? For example, the peroxisomes for beta oxidation. And this is a question from Regan Miller.
Yeah, thank you for the question. This really differs depending on and some of the gene expression profiles that they express. So some cell types are more sort of prone to accumulation of lipid droplets, and there's also differences, for example, in the expression of transporters for the uptake of exogenous fatty acids and the accumulation of these lipids in lipid droplets. And in addition to their sort of propensity, basal propensity, depending on their expression profiles, they're sensitive to signaling pathways. For example, they're beta-adrenergic pathways, which are one of the best characterized, that induce phosphorylation of lipid droplet proteins and lipolytic degradation of lipid droplets, freeing up triacylglycerol and fatty acids that are stored in lipid droplets. So I think it's going to be a combination of the proteins and expression profiles of those cells, as well as the metabolic conditions and the hormones and signals that those cells are exposed to.
All right. Thank you, James. This was great.
Thank you, Eric, and thanks everyone for attending and for the questions. Hope everyone is doing well. Take care.
Thank you.